All right, I'm not even sure the people online can see this, but I'll try. All right, so pulling up the lab. Evolution lab, DNA lab. Okay, here we go. So if you pull it up, if you hover over the video, it brings you to it, as O'Brien pointed out to me. Like if you hover over that, it kind of works like that. See? On this one, it doesn't do it. So all you have to do is copy it like that, right click, copy. Then we'll go up here and open a new tab, paste it. Come on. There we go. And then it brings it up.
Okay.
I mean, don't get me wrong. I will still print it for you if you want, but you can. That's how you'll do it, is a copy and a paste.
Okay. I'm gonna try it right now.
Why I love questions. You think you might be the only one with that problem, and then it turns out there's a bunch of people who need to know these things. So thank you both.
Mr. Reed, would UK essays be a literature cited for the lab report?
Would what be a literature cited?
UK essays.
Sure. I'm very, very not strict about literature cited. The reason I have it is because it's kind of like a university standard. And also, if you put in something that's incorrect, I'll check that first. Like if you say, photosynthesis produces oxygen or produces carbon dioxide, my first reaction would be give you a point off. But if I check your source and your source is just wrong, I would tell you like, hey, your source is wrong, but you wouldn't lose the point because you backed it up with a source. Good morning, Johnson. And good morning, Lloyd.
Good morning.
Okay.
Did it work for you?
No. It says, it looks like there aren't many great matches for your search. Try using a word that appears.
So did you copy the exact thing?
Mm-hmm. Let me do it one more time.
Let me watch as you do it. You have to be careful not to copy the part right before it, because the part right before it says.
I think it's this one right here. I'll type it in Lex before that.
Oh yeah. See, I'll do the part where it starts at HTTPS, not the part before that. There you go. Yeah, that should do it. I hope. But again, if not, let's print it off for you.
Yeah, I was on the tear Friday. Getting my work knocked out. And I did this lab. I was so excited. Yep, they popped right up.
Sweet.
So I'm gonna leave this like this until later on.
All right.
And that natural selection lab, that was fun.
Oh, you already did that one? Awesome.
With the white and black box. Just like I thought, if something white would stand out on something dark, and something dark will stand out on something light. So I figured I will catch more of the opposite.
Yeah.
And that's the way it worked out.
Yeah. I think it's a great example of natural selection. One, because it's fairly easy to understand. Like you said, hey, light moths stand out in a dark environment. So obviously they're the ones that are going to be eaten. Therefore, it's going to be the dark ones that survive, reproduce, and pass along the genes. And another good thing I love about it is that it's happened recently. So a lot of times when people say they don't believe in natural selection or evolution, usually what they really mean is they don't believe that that's how humans came to be. And that's a whole different discussion. We don't even discuss that in this class. But obviously we can see that natural selection, the theory, is a real thing. Again, you can argue all you want the hypothesis of the humans coming from this ancestor versus that ancestor, but completely different discussion.
Yeah, first with the moth thing I was talking about.
Yeah.
I was confused at first of the play game on there. I was confused there. I'm like, okay, because I watched how to play. I'm like, just moving my cursor, just wondering what to do. I'm like, okay, there's a timer. What am I supposed to do?
Because it was hard to see it, right?
Yeah. So then I clicked. I'm like, oh, okay. Let me just quickly, because it was like 30 seconds. I'm like.
Well, that's okay. And here's a little hint for you guys and anybody online and anybody watching the video. I have no way of knowing if your numbers are correct. The only thing I look for is make sure they all add up to 100%. So, if you've got like 50% and 60%, you're going to get it wrong. But if you have 40% and 60%, then that adds up to 100, and I'll take it. Let's see here. Good morning, Ray.
Good morning.
If it's only going to be two of us.
Yeah. Wow, yeah, it's already 7:58. It's a case of the Mondays. Well, yeah, if it's only going to be two of you in here, then you guys will get even more extra credit for being here. I'm actually going to look at something. I'm going to average up the attendance.
Is the lab still going to be half credit if it's overdue?
Yes. Well, depending on it, the improvements will be half, so like anything that was worth a point that you fixed, you're going to get half a point back. Good morning. Tyrus is here. All right, well, I guess I'll start attendance. Since it's 7:59, we have a little bit less than a minute. And we are a little bit behind, so I'm going to try to move quickly here. Before I start attendance, I'll remind you guys online, make sure you pay attention to what we're doing, because just because you're here right now and I'm about to call out your name, or I already have called out your name, does not mean you are good for attendance points. Just a reminder. Speaking of which, we'll do what we did last time. We'll do a series of words throughout the video, throughout the lecture. And when you email me at the end, you'll just have to give me those series of words. And the first word in the series is clock. So make sure you guys heard that. The first word that you're going to email me for attendance is clock. Anyway, Sullivan, not here. Hill, not here. Byrne, not here. Tyrus is here. Lacey is here. Thornabar is here. Fitzpatrick is not here. Hamlet, not here. Huffman, not here. Johnson is online. Bayard, not here. Sigmund, not here. Pena, not here. Michael Baumgardner is here. Wow. Right on cue, Michael. Ray is online. O'Brien, excuse me, just to be clear for my own recording purposes. Michael Baumgartner is here online. Ray is online. O'Brien is here. Hamrick is not here. Lloyd is online. Davis is online. Elias is not here. Breeden is not here. Maxwell is here. Let me go back down. I see Fitzpatrick came while I was doing this. 1 second, bear with me. All right, Fitzpatrick is online. All right, did I miss anybody? I see Davis, Johnson, Lloyd, Ray, Fitzpatrick, Baumgartner online. I don't think I missed anybody, but just to be sure, everybody's accounted for, right? Okay, so quickly with announcements. So independent work is done. Hooray, it's one less thing to worry about. Obviously, I have a lot to grade, and I find it, or when I was a student, I was always not upset. I didn't like it when people took forever to grade and return assignments. So I'm apologizing in advance. It's going to take me a while to grade all these because a lot of people waited until the last minute to turn things in. So it's out of my control. I'll do the best I can. I'll remind you that independent work, the maximum number of points is 100. At least half of your points needed to come from written work. All that being said, that's the official answer, right? So some of you have been done for a while. But any points you get above 100 officially does not count, right? Because 100 is the max. But if at the end of the semester I don't like the grades, like it's a 100 level class, and if I feel like I've got, I don't know, too many C's and not enough B's, then instead of bumping it or curving it like most some professors would do, I'm going to hit a button in my spreadsheet, and then all those extra points will count. So rather than being like, all right, this person has the highest C, so they're the ones who's going to get bumped up to a B. I'm not going to do that. I'm going to hit a button, and then I'm going to count all your extra credit points, whether that be for attendance or independent work. So what I'm getting at is feel free to keep doing the other extra written work. I'm not going to grade any more the other independent work. I'm not going to do any more written work, but you can still do videos. You can still do study guides. You can still respond to my emails, which I don't know why some of you haven't done it. For the past week or two, I put out an email and said, hey, if you read this, reply to it for free points. And some of you still haven't done it. Read my emails. Anyway, you can still do that stuff. Officially, it won't count. Unofficially, it might at the end of the semester. What else to say about independent work? Yeah, I think that's it. Yeah. So again, if I were you, especially if you're worried about passing, if you have the time, it wouldn't hurt to continue to do some independent work, at least the stuff that really helps you get ready. Like go back and take exam 2. I said that when I released the grades. I said, if you redo exam 2, those extra points will go towards your independent work. That still stands, and it'll help you. Redoing exam 2 will help you prepare for the final exam. Anyway, any questions about independent work now that it's past due? All right, that's pretty straightforward. The lab, I'll remind you, this week is in person. Even though it's an online lab and you need to bring in your computers, your devices or whatever, it's going to be in person. You're going to stay until 1:50 or until you're done with your lab, whichever comes first. And again, if you've had it done and I've given you your grade back, then you don't need to come in, right? If you've done it, it's been graded, no need to come in. I've had 2 students tell me there's issues with the lab. When you click the link to take the questions that go along with the video, it doesn't work. So you need to copy that link and paste it. And if you don't know how to do that, if you watch this video, and I know you guys are here online live, but if you go back and watch the video before class started, I showed you guys how to do it. So just watch that video if you don't know how to do it. Any questions about lab or anything else? All right. So now that we're done with independent work, I highly recommend you guys start working backwards. If you have absences, look at the grade sheet, see which days you missed, and then go back and watch the video, do what you need to do to get your points, or at least half points. If you are missing assignments or did poorly on labs, go back and fix it for half credit, right? You can still fix your stuff. And now that we've passed independent work, now's the time, in my opinion, to start working on old material. All right, good timing. We're just now done with announcements, so let's jump into chapter 9. So Elias is here in person. Any other questions or any questions before we get started? All right. Well, let's get started then. To remind you, we are still in chapter 9. We should be done with it, but here we are. We ended with talking about incomplete dominance. And before we go back into it, let me remind you where we're sitting. Prior to this middle bullet point, right? Almost all of last week, we were working on this bullet point. Excuse me. Let me undo that. Well, I can't. Anyway, prior to last week, we were working on this bullet point right here. Or excuse me, most of last week, we were working on that top bullet point. And in that top bullet point, we were talking mostly about things that were controlled by one gene, right? And there was only two phenotypes. It was either this or that. It was either the dominant or the recessive phenotype. Then, towards the end of the chapter, the end of the week, last week, we started getting into these variations, right? And the first variation that we've talked about, which is what we ended with last week, was incomplete dominance. So in that case, it's not this phenotype or that phenotype, right? It's not necessarily dominant or recessive. It's a blend of both. So the example I gave, or that your book gives, is red and white. What happens? First of all, how many of you are art majors? Just the one? So you don't answer this because you're going to know it. The rest of you, it's a hard one. Think hard. What do you get when you mix red and white? Pink. Geniuses. Yes, pink, right? And that's incomplete dominance because you're mixing the two phenotypes, right? Another example is if you had, I don't know, a blue flower and a yellow flower, and then you get a green flower, right? That's because you're mixing the two. So again, that's the same thing. And just to remind you, you still have the 1 to 2 to 1 genotypic ratio that you see down here at the bottom, right? 1 to 2 to 1. You still have that same genotypic ratio when you cross the heterozygotes, except now the phenotypic ratio is different because you have one dominant right here, right? You've got one recessive, and then you've got these two that are the mix of the two. Your book gives you another example, and we didn't talk about this. So here we go. We're jumping into new material here. The other book example is hypercholesterolemia. All right, that's a good human example. Forget about flowers. Let's talk about something that affects humans. This is when you have dangerously high levels of cholesterol. Your books, and you don't need to necessarily write any of this down if you don't want to, because this is just an example. I won't ask you any questions about it on the exam. But heterozygotes have about twice the normal level of cholesterol in their bloodstream, which isn't good, right? And that's just heterozygotes. So that would be that example of the incomplete dominance. It's not recessive, it's not dominant, it's that blend of the two. Now homozygotes, they have about 5 times the normal. So because of that, people who have this hypercholesterolemia that are homozygous, they have what it says right there, heart attacks as early as the age of 2. So imagine having a little 2-year-old toddler running around with having a heart attack. That hurts my heart to think about it. The next slide I'm going to show you kind of visualizes how it happens. And again, I'm going through this quickly because we're behind and because I'm not going to ask you any questions about this on the exam. But here's what it looks like. So here's a normal cell. This person, they are homozygous dominant, meaning they've got this gene codes for this protein. There's a protein on your in your cell surface, in your cell membrane, that takes cholesterol out of your bloodstream, right? So this person has got 2 alleles that work for it, right? So because of that, it's making a lot of protein, the normal amount of protein. Therefore, you're taking the normal amount of cholesterol out of your bloodstream. Here's the second one. Someone who's heterozygous, they've got this one allele that says, all right, this is how you make the protein that takes away cholesterol. This person's allele is just broken, right? So they still have the gene, but it's broken. It's just a defunct, if you will. It's a gene that doesn't work. So because of that, they're only producing half the amount of proteins. Therefore, they're taking out less cholesterol out of the bloodstream. And this person who is homozygous recessive, they have 2 alleles, neither of which work. So they're not producing any of these proteins that take out the cholesterol. Therefore, they have a lot of cholesterol in the bloodstream. Makes sense, right? This is a good time to point out an independent work topic if you're interested. You can look into it. It has to do with evolution. It's interesting. If you look this up, I'm going to give you one example, and you can look up to see if there's other examples. Humans are one of the few mammals that can't make our own vitamin C, right? Most mammals can make their own vitamin C, humans, and then primates cannot make our own vitamin C, which is interesting because if you were to look at it, it would probably look a lot like this. What I mean is Humans have the gene to make vitamin C. It's just that it's broken, which is interesting to me because that means that indicates that previously there was an ancestor in which the gene wasn't broken yet and we could produce our own vitamin C. So you can look into that and write more about it if you want, or see if there's other examples of that. Here we go. So we're done talking about incomplete dominance. The next thing we're going to talk about, even though your book presents it as ABO blood groups, which is weird because that is just an example of what we're about to talk about. What we're about to talk about is codominance. I'm going to abbreviate because it's really hard for me to write like this. So yes, ABO blood group is the example that your book gives. I don't know why it gets its own bullet point, because really what we're talking about is codominance. Oh yeah, and it's also an example of multiple alleles. So to remind you, so far everything we've talked about has been, dominant or recessive, right? There's the dominant allele or there's the recessive allele. There's only been 2 alleles. And then when we talked about incomplete dominance, I did provide you with a third phenotype because it was a mix of the 2. Now we're getting a little bit more complicated, and we're talking about multiple alleles, 3 alleles to be exact, and also codominance instead of incomplete dominance or complete dominance. You don't necessarily need to write this down either because, again, it's just an example and because we are behind, so I'm going to move forward. I'm going to show you a picture. All this information that's on the slide is on the next slide, and it'll make much more sense when I show you. Here we go. I'm just going to explain how this works. You all know blood types. I'm sure you've heard of them. Your blood types are either A, B, AB, or O. And the thing is, there's 3 different alleles. So in this case, maybe I'll laser pointer. You guys tell me if you can see this because I don't want to get up and block the camera. Can you guys see where I'm pointing at here? Can you online see where the laser pointer is?
Yeah.
Okay, good.
Yeah.
Thank you. So if you look here, let me draw it first. The different genotypes they all have an I. So there's 2 different dominant phenotype or genotypes, 2 different dominant alleles. There's I with a big A here, and then there's also I with a big B, right? That's the other allele. Those are both dominant. And then the recessive one is just a little I, right? So now we're dealing with 3 different alleles instead of 2. That's the first thing you need to think about. Now let's look at the different groups here. If you are blood type A, what that means is you have at least one of these dominant alleles, right? The I with an A. You have at least one. As long as you have that one, then you've got the gene that says, hey, produce this thing. So because of that, you have these carbohydrate A on the outside of your blood cells. So again, if you have at least one of the A alleles, that means you're going to produce the A carbohydrate. Now here's where it gets interesting, or at least relatively interesting. If you have the A carbohydrate, that means your blood recognizes B as foreign. So you cannot have B blood. If it sees anything with a B in it, it attacks it. And that's why you have to have the right blood type. Specifically, again, because you're anti-B, obviously you can't have B, right? If you have B, you're anti-B, so you know that's going to work. But here's where it gets a little bit more interesting. Again, you also can't have AB blood type because even though you are A, you were so anti-B that you won't take AB blood type. Obviously, you can see here, you can take A blood type, which makes sense because that's your same blood type. One part that's a little bit less intuitive is this whole thing about the O blood type, which is why I'm going to skip to the bottom and then come back up. If you are O blood type, that means you have this. You've got these 2, you're homozygous recessive, meaning you've got 2 alleles that don't even produce carbohydrates. So on the outside of your blood cells, you have no carbohydrates. Therefore, because your body is basically doesn't produce the carbohydrate, it recognizes any carbohydrates as bad. So you can't take any blood type other than your own. The interesting thing about the O blood type is that because you don't produce any carbohydrates, anybody can take your blood, right? Because blood type A is anti-B. Well, you're good because there's no A or B. Blood type B is anti-A. Well, O doesn't have any, so it's good. Then that brings us to the last one I'm going to talk about. You'll notice I skipped B. Who cares? Blood type AB, that's when you have 2 of these dominant ones. You have the A allele and the B allele. Therefore, you're producing both carbohydrates. Therefore, you're not anti-anything. So you can take any blood. So even though I'm not going to ask this, and the study guide might, someone might ask you, what is the universal donor? And that would be O, right? Because anybody can take the O blood type. And then someone might ask, what is the universal receiver? And that would be AB, because AB can take anything. But any questions about this? Again, it's just an example. I'm not going to ask any questions about it. I'm even going to skip the next slide. It just talks about how if you have the wrong blood type, your body attacks it, and that's why it's deadly. That's why it's not good for you to have the wrong blood type. I've already discussed all this. I've just put it into words right there. Same here. We've already talked about this. Now I'm going to give you a better example. Here's the example you need to know. All right. Not specific. You never need to know a specific example, but here's the kind of things you should think about when we talk about codominance. So again, before we talk about this, let me back up and remind you of what we talked about so far. Complete dominance. You either have this allele or that allele, right? So you're either chimple or good grief, chin dimple or not, right? Widow's peak or not. It's either or. Purple flower or white flower. That was complete dominance. Then earlier today and at the end of last week, we talked about incomplete dominance, which is the mix of the two. If you mix white and red, you get pink. Right? And you can see that here. Now, codominance, it's a lot like incomplete dominance because you do see both of those phenotypes, except now they're not blended. Here's what I mean. I like this picture here. With this dominant situation, right? If you were to have if red was dominant to white, and you were to cross a red and a white flower, purebred, then all of the offspring would be red. That's complete dominance. Then if you were to have this incomplete dominance, this was what we talked about earlier. You mix red and white, you get a blend of the two, which is pink. Now, this is what we're talking about with codominance. You see this is a red flower with white spots. You guys see that, right? Everybody online can see that? All right. And another really life example is if you mix a white cow and a red cow, it's not like an orange cow. What happens is you get a white cow with red spots, right? You see each individual phenotype, both. They're not blended. They're both there. This is a good time to tell you how this exam is going to go. I'm going to give you a situation. I'm going to say this hypothetical thing breeds with this hypothetical thing, and these are their offspring. What is that? What kind of dominance is it? And if I might say like a red alien mates with a blue alien, and 100% of the offspring are purple, then you would say that is incomplete dominance, right? Because that's a blending. When you blend red and red and blue, you get purple. Now, if I say you have a red alien and a blue alien, and 100% of their offspring are red with blue spots, then you would say that is codominance because both phenotypes are there. You guys understand? There's almost going to be no questions where I ask you what codominance is, what incomplete dominance is. Most likely, I'm just going to give you a scenario, and you need to recognize it as one of these things. So are we good there? It's pretty straightforward. I remember when I was a student, the hardest thing for me to remember was the difference between codominance and incomplete dominance because they're so similar, and even the names are similar. So you just somehow have to remember incomplete is blending and co is both. Maybe it'll help you think about co-parenting. Does anybody know what co-parenting is? Not in biology. Like if you're talking about a divorced couple with a kid, what is co-parenting? Yeah, they're both sharing responsibilities, right? So you see some mom and dad, right? They're both doing things. Or maybe the kid lives with the mom and the dad. So it's not a blend, it's this and this. You know what I mean? Usually co-parenting doesn't mean the divorced parents are still living together, blended. It's like there's mom house and there's dad house, but the kid is utilizing both. Maybe that'll help you remember. If you come up with another way to help you remember, please let me know. I'll give you extra credit for it, and I can use it moving forward. Whew. So any questions about codominance? All right, here's another one. The next one, it's a lot easier than the name sounds. Pliotropy. That sounds like a weird name, but it's actually pretty simple. So again, I want to remind you, so far everything we've talked about, that we've seen some variations, but thus far it's always been one trait or one character, right? So we've either been looking at like just the flower color or just the chin dimple or just dwarfism or not, right? It's always been one character. Now we're going to look at something a little bit different. Pliotropy is when you have one gene that affects several characters. it would almost be, and this is definitely not the case, but it would almost be as if that lab we did where we looked at chin dimples and bent fingers and hitchhiker's thumb and tongue rolling and all that. It would almost be as if there was one gene that controlled all of those things. And of course, there's not. Your book gives the example of sickle cell disease. And normally I don't ask questions about examples, but sickle cell is something we're going to talk about a lot. We're going to talk about it in the context of pleiotropy. Next chapter, we're going to talk about it in the context of mutations, because we're going to talk about DNA. And then in the next chapter after that, in evolution, we're going to talk about it in the context of how certain humans are more prone to have sickle cell disease than other people. But anyway, let's talk about it very quickly. Sickle cell disease exhibits pleiotropy, meaning there's this one gene that causes sickle cell disease. And you could almost argue that that's not pleiotropy, right? You could say, well, the one gene just gives you the one disease, right? And if you were just going to consider sickle cell disease itself as a character, then you'd be correct. However, this disease has a lot of different characters that come along with it. How does it happen? Well, the gene, or the mutation in the gene, causes an abnormal hemoglobin protein. That'll be more important on the next chapter, so I'm going to skim over it for now. But that abnormal hemoglobin protein, remember, proteins are all about the shape, right? So because it's an abnormal shape, it causes the red blood cell to also have an abnormal shape. You can see right there, it's an abnormal shape. Has jagged edges. Can't really see it in that picture. Point is, though, remember form and function, right? Because it has the wrong shape, its function is going to be messed with. So your book points out that it's usually only the homozygotes that have the disease. I'm not going to ask you that detail. So far, the only detail you need to know on this slide, if anything, is that sickle cell disease is a great example of pleiotropy. One gene, many characters. That being said, we haven't got to the many characters part yet, so let's jump into that. So what happens? An individual who is homozygous for the sickle cell allele, meaning all their genes are saying, hey, this is how you produce this protein, right? This hemoglobin protein, and they're all messed up. Both versions, you've got both alleles are messed up. So therefore, all of your proteins are going to be messed up. Therefore, you will have sickle cell hemoglobin. Therefore, this hemoglobin is going to crystallize into these long, flexible chains that causes the red blood cells to get this weird, jagged shape. You can see it's not like the round donut shape that you're used to. And these sickled cells can lead to a cascade of symptoms such as weakness, pain, organ damage, and paralysis. You do not need to know any of these examples, any of these things that are listed. The point here, and the thing that you might need to know for the exam, is that sickle cell disease is a great example of pleiotropy because it's one gene, many characters. Oh, this reminds me of another independent work topic, and you can back this up to a few things we talked about today. I'm giving you examples of complete dominance. I'm giving you examples of incomplete dominance. I'm giving you examples of codominance, and I've just given you examples of pleiotropy. So for anything I give you examples for, you can do it. Oh, never mind. What am I thinking? Independent work's done. I got to get out of this rut. Never mind. Forget it. You can look it up if you want. You can look up some other examples of pleiotropy if you want, but you can even write about it and send it to me, but I won't grade it. All right. The next thing we're going to talk about. Again, this sounds a little bit complicated, but it's not. Just like pliotropy, unless you've ever heard that word before, it might sound complicated, but it's not, right? It's one gene, many characters. That should say polygenic, not polygenetic. I'm going to skip ahead. Polygenic inheritance is the next one we're going to talk to. So in this case, it's kind of like the opposite of pliotropy. It's not one gene, many characters. It's many genes that add up to one character. Specifically, as your book words it, the additive effect of two or more genes on a single phenotype. Blah, blah, blah. Just remember, multiple genes, one character. That's the way you should think about it. Again, like I said, it's the opposite of pliotropy. The way you would recognize this, and this is the way the question is worded in the study guide and possibly on the exam. The way you would recognize this is that if you were to graph the different phenotypes of a population, it would be a bell curve, and that'll make sense when I show you the picture. And I'll give you an example of what it wouldn't be, right? So if we were talking about widow's peak. All right, widow's peak. If you were to graph the whole human population, and I don't know which is which, that it would only be 2 bars, right? You either have widow's peak or you don't, right? So there'd be a bar, like, I don't know, maybe 40% of the people have widow's peak and 60% of the people don't. I don't know what the numbers are. But that's what it would look like, right? There'd only be 2 bars for the entire human population. You either have it or you don't. If you were to graph those flowers we were looking at, I don't even know what species they were. If we're talking about incomplete dominance, and you were to look at every single animal, every single one of those species on Earth. Let's see, it would be either white, pink, or red, right? It would only be 3 bars, right? No matter what, because there's only 3 phenotypes. You with me so far, right? So far I've shown you a graph where there's only 2 bars. Here there's a graph where there's 3 bars. This is what I'm getting at when I'm talking about the bell curve. If you were to graph height, for example, and this is still a very simplified version. Height is obviously much more complicated than this. But if you were to graph height for the whole human population, there wouldn't be 2 bars, right? Because it's not like everybody's 5 foot or 6 foot, right? There also wouldn't be 3 bars because it's not like everyone's either 5 foot, 6 foot, or 5'6", right? There's more than 3 different heights. There's a lot. So if you were to graph anything that has multiple genes for the one character, you would get a bell curve. And again, this is a simplified version. This is only showing you, this is still, remember the Punnett square? This is the Punnett square. It's not the easy one that you saw that looked like this when we did monohybrid crosses, right? That was the monohybrid cross right there. It wasn't even the dihybrid cross, which was a little bit more complicated at a 9-to-3 to 3-to-1 ratio. This is one with 3 genes. Not even alleles, but right, but 3 different genes. And you can see the ratio is a lot different. Here you would have a 1-to-6 to 15 to 20 to 15 to 6 to 1 ratio. And again, that's only 3 genes. This is a very simplified version of what height would be. There's many, many genes, and not to mention environmental effects. But anyway, I guess what you need to know for the exam, the most important thing that you need to know is that polygenic inheritance means that there's multiple genes affecting one character. And the other thing you need to know is, like, the way you would identify it is if you were to graph, a bar graph of all the different phenotypes in a population, you would get a bell curve. Or even if you want to shorthand that for grading purposes, polygenic equals bell curve, because that's the only time you're going to put a question on the exam where it mentions anything about a bell curve. All right, any questions about this? All right, so like I said, with height, again, that's just a simplified version because there's more than 3 genes. And what's the other thing I said about height? It's also not just genes, it's also your environment, right? So you might have 2 identical twins that have the same exact genes, but if they were separated at birth, like maybe because of the foster system or whatever, and one of them's growing up in poverty and they're malnourished, and the other one's getting all the nutrients they need, then when they grow up, they're probably not going to be the same height. Those identical twins with the same exact genes, one's probably going to be taller than the other. And that brings us to something called epigenetics. Epigenetics. Who knows what epidermis is? Skin, right? And not the whole thing, right? The top layer, right? So you can think of it's like above, right? It's almost like the above part of the skin. In epigenetics, you can think of it almost as the same way. So we're no longer looking at just the genes, just the DNA, right? That code written. We're looking above that, so to speak. So many characters result from the combination of heredity and environment. Of course, I'm not going to ask you that. How many? I'm not even telling you. I'm just telling you there's many. We don't know how many. It's actually a topic of debate whether some things are influenced more by genes or environment, right? If independent work wasn't done, that'd be a very, I think, interesting thing to talk about. What about aggression? The biggest one that I used to hear a lot about was homosexuality, right? It's like, is that nature or nurture? Is that in your genes or is it in your environment? And your book points out, and I think it's a great example, spending time with identical twins is convincing that the environment and not just the genes affects a person's traits. Because again, identical twins have the same exact genes, unless there was some sort of mutation. So even though they have the same genes, they are slightly different because the environment is acting along those genes in a different way. And I think I mentioned at the beginning of this chapter, you can think of genetics, you can think of chromosomes like books of instructions, right? Books of instructions on how to build stuff, specifically proteins. But imagine, you have a book and you're trying to build a protein, and you got twins here. They've got the same book, the same set of instructions. But I go to this twins book and I try to open it to whatever page because I want to build something, and it's stuck together, right? I can't even get to it. So I'm spending all this time just trying to get the page apart so I can read it. And then finally I get to it and I can make a few of those products. This other twin, I go there and that book's wide open, already the page I need. I can build a bunch of whatever it is that I need built, right? So even though these two twins have the same exact set of instructions, I can build a lot for one twin and I barely build any, or if at all, for the other one, simply because of the way the book was open or shut. And that's how epigenetics is. That's how you should think of it, basically. Your chromosomes, yes, they may have the same genes if we're talking about identical twins, but they're not always as easy to read, right? Sometimes the chromosome is really easy to read. Sometimes it's hard to read. Because remember, we talked about this when we talked about DNA being wrapped around histones. Or was that next chapter? Anyway, we'll talk about it definitely in more detail next chapter. But the genes are not equally read, is basically what I'm saying. And also your environment turns different genes on. For example, when you drink milk, that kicks on some genes where, and you have learned about this word before, lactase. Remember, that's the enzyme that breaks down lactose. Your body's not making a bunch of lactase until it needs it. So when you drink milk and lactose comes into your system, your body says, okay, I'm going to make some lactase. That turns on those genes. So again, if you have two identical twins sitting here, one's drinking beer and one's drinking milk, it's only the one who's drinking milk that's going to turn on those genes to make that lactase. So anyway, yeah, nothing necessarily important on this slide. This is kind of what I was saying earlier. You don't need to write any of this down. Remember I was saying with the chromosomes, sometimes it's easier to read them. Sometimes it's harder to read them. Your book gets very specific with this bullet point right here. Chromosomes can be chemically modified by adding or removing chemical groups on the DNA and or the protein. Because remember, a chromosome is half protein, half DNA. So if you mess with that stuff, it's going to be harder or easier to read the gene. There's literally a machine that clamps onto your DNA and reads the code. So you can make it harder to do that, and you can make it easier to do that. That is epigenetics. The environment does play a role in those changes. Your book also points out that this may explain how one identical twin can suffer from one genetically based disease, whereas the other twin doesn't. So again, both twins have the disease or the gene that says make this disease, to simplify it. Well, for one twin, that gene that says make this disease, again, the simplification, just can't be read. The gene's there, but it's not being read. The disease is not there. The other twin has the same gene, but the DNA is being read, the proteins are being made, the disease is shown. Anyway, finally, this last bullet point here, simply put, epigenetic changes are changes to the DNA and the chromosome, right? So you're changing the actual chromosome itself and how it's read, but you're not changing the actual code, which is the A's and the T's and the C's and the G's that you're going to learn about in the next chapter. Anyway, or even simpler put, for the exam, what you need to think about with epigenetics means it's not just genes, it's also the environment. So if we talk about environment influencing genes, you should think of epigenetics. And to clarify, I want to clarify because I doubt anybody will think this hard about it, but just in case, lactose, lactase, that whole thing, that's not really a great example of epigenetics. That's just an example of the environment turning on and off a gene. It was a good example for our level. So don't go quoting me on that with epigenetics and lactase, but there's a good way to think about it at a 100 level class. So any examples? All right, we're almost done with epigenetics. Your book also points out that these modifications, right, that you're making to the chromosomes themselves, as opposed to the actual letters of the gene, they can be carried out, but they can also be reversed. So this is also an interesting thing to think about, because if you cause mutations, I don't know, we'll talk about that next chapter. Next chapter, we're going to talk about mutations. And when we do that, then I'm going to also back it up and bring this idea up again, this whole idea of epigenetic changes that can be passed along. Because it used to be, people thought that you could only pass along mutations, like when you change the code, used to think that was the only thing that can be passed down, but we also learned that now these modifications can also be passed down. So any questions about epigenetics? All right, the next bullet point is really, really quick and easy because of the way your book presents it versus the way I present it. So this whole time, as I've been presenting things, I've been talking about chromosomes. I've been talking about how in meiosis, these alleles split up. So I've already said all this, but your book at the end, it gives you all this information from these first two bullet points, and then it comes back and says, okay, now that we've said all that, let's look at this from the point of view of the chromosomes. And I've already done that. So again, I'm going to go through this quickly because we've already discussed most of this, but now we're going to do it strictly from the context of the chromosomes. So the chromosome theory of inheritance, it states that genes are located at specific locations on chromosomes. You already know that. They're called loci, or locus would be the singular. We know that the behavior of the chromosomes during meiosis and fertilizations account for these inheritance patterns. So again, the alleles splitting up and then coming back together. We already learned about that. And we also know that it is the chromosomes that undergo segregation and independent assortment. See, we've already talked about that, right? And these are the things that accounts for Mendel's laws. So we know, again, segregation, right? So the alleles separate, right? You only have one allele from each of your parents. Any gene you talk about, unless there's a mutation, you only have one allele. That's segregation. Independent assortment. Remember, when all your cells line, or all your chromosomes line up before you produce a sperm or an egg, it's not like all your mom alleles and chromosomes are on one side and all your dad are on the other, right? For all 23 pairs of your chromosomes, it can be any which way. It could be all mom on one side and all dad on the other, but we don't know. It's all random. That's independent assortment. And anyway, again, like we've already said, these are the things that accounts for Mendel's laws, which we've been talking about the whole time. So here we go. Here's something you've already seen, but now your book threw some chromosomes in there. If we're looking at seed, color, and texture, right? The color, is it yellow? Is it green? The texture, is it smooth or is it wrinkled? If we look at that, because they're two different genes that are inherited separately, there's two different chromosomes we're talking about. I don't even want to talk about this too much because we've already talked about it. But basically, again, because this is homozygous dominant here, this one's homozygous recessive. Yes, when this thing goes through meiosis, they're going to split. But look, this thing only has big R's and big Y's. That's all it can offer. This thing only has little R's and little Y's. That's the only thing it can offer. So the only possible outcome for these things, and you should recognize this as a dihybrid cross, only thing that would be possible is this heterozygote, which is big R, big Y, little y. This is things you've already learned. This next picture is also this, but we're going to expand it. You've already seen this, except now we're just giving you pictures of the chromosomes instead. Yeah. So I'm going to go move past this because we're running behind on time and we've already talked about this. Let's expand this even more. If that wasn't too much information on the slide, there's some more. But again, the point is, with all this, again, with this dihybrid cross, you should still recognize this 9 to 3 to 3 to 1 ratio, the phenotypic ratio. And again, instead of just saying, hey, here's the phenotypes, this picture actually shows you how it works out with each individual chromosome. So you can study that if you want, but we've already talked about it. I'm not going to ask you any specific questions about it. So everybody good there? All right, the next one's linked genes. We haven't talked about these, so I'll slow down a little bit for this. Linked genes. What do you think that means? Any guesses what linked genes might be? Obviously, they're linked, but how are they linked? No guesses? All right. You'll need to know this for the exam. They are located near each other on the same chromosome. That is how they're linked. Meaning, you know, previously we were talking about how things are not inherited together, right? The example we just used was P color versus the P texture, right? Yellow or green was one character, or two, you know, the color was one character, yellow and green being the traits, and the texture was the other character, right? Smooth or wrinkled. Those things were not inherited together. They're on completely different chromosomes. So you could be yellow and wrinkly, or you could be yellow and smooth. You could be green and wrinkly or green and smooth, right? Because those two things had nothing to do with each other. So you could have, again, your mom allele for the color, and then your dad allele for the texture. Linked genes are not necessarily, they don't necessarily follow that pattern because, again, they are close to each other on the chromosome. Therefore, if you got, let's look at this picture right here. If you're looking at gene 2 and 3, whatever it's for, and this is not the case, but let's do this because you're familiar with these phenotypes. Let's say gene 2 was for chin dimple, right? And let's say, all right, gene 2, chin dimple. Let's say gene 3 was for, I don't know, widow's peak. That's like saying, because they're so close together, if you got your mom's allele, let's say this is your mom's chromosome. Let's say you got your mom's allele for chin dimple, right? Let's say, I don't know, maybe she's big D, just dominant. And she also happened to have little W, which is recessive for widow's peak. Whatever you got from your mom for the chin, you probably got it from the widow's peak. Because in this hypothetical incorrect scenario, they're right next to each other, right? So if you're getting, obviously, if you got your mom chromosome, then you got all those different alleles that come along with that chromosome. Does that make sense? Does anybody have any questions? What I'm saying, again, they're not independent anymore, like we learned about previously when we talked about independent assortment. Yes, all the chromosomes, the mom and the dad chromosomes line up independently of each other. There is no rhyme or reason. But if you inherit the mom chromosome, generally speaking, you're going to inherit every single allele from your mom, from that chromosome. That being said, there are some exceptions because we have something called crossing over. And I'm not going to get into too much of this. You do need to know about crossing over, but we've already talked about that. But keep in mind that even though, again, if you inherit the mom chromosome, you inherit every single allele that comes with that chromosome, right? But the exception to that, something just keep in the back of your mind, not even necessarily for the exam, just to keep in the back of your mind. In real life, if you've got this dad chromosome right here next to it, and it's got a beer belly, a dad bod. Anyway, it could, remember, in prophase of meiosis 1, maybe this part from this line down, maybe they swap, right? You might have crossing over. So in that case, you might have a mom chromosome with the dad chunk down here. And here you would have a dad chromosome with a mom chunk down here. But generally speaking, again, if it's on the same chromosome, then they are inherited together. And that's something you could, independent work's done. Something you could look up if you're interested, but you don't have to write about it. There are examples, human examples of linked genes, where if you get one from your mom or your dad, you probably got the other one from the same parent. Anyway, let's move forward. Here's a really, really easy one. I almost want to cry every time someone gets this wrong in the exam. As I'm teaching this, I'm thinking this is one of the easier things that I teach. So here we go. Buckle up. We're talking about humans, and we're talking about sex determination. What makes you a male? What makes you a female? And we've already said it in the last chapter. We're saying it again. So I don't know how anyone's going to miss this, but they will. Here's what you need to know right here. Males have an X and a Y. Females have an X and an X. That's it. Please don't get that wrong. Please. Such an easy. Females are XX, males are XY. If you're online, the second word that you need to give for attendance is going to be a microscope. So when you email me afterwards, as of now, you have 2 words, and the second one is microscope. If you missed the first one, you can watch the video. Any questions about the word or sex determination? It's pretty simple. XX or XY. All right. There's a picture of it. So if I were to give you this picture on an exam and say, is this a female or a male? What are we looking at here? Good, a male, because it's an X. That part's the same as a female, but there we go, a Y. That's what makes you a male, right? The Y chromosome. That brings us to sex-linked genes. A gene that is located on a sex chromosome is called a sex-linked gene. Mind-blowing, right? That's what a sex-linked gene is. So it's any gene that's on a sex chromosome as opposed to any of the other autosomes, right? Your book also points out, and I probably won't ask you this, but most sex-linked genes are found on this chromosome, to the left, and not this one. Most on the X and not the Y. Any guesses to why that might be? Don't overthink it. Just look at the picture. Why would you see more genes on the X chromosome than the Y chromosome? It's bigger. Yes, that's it. Again, think of this as a set of instructions, right? If someone gives you a book this thick, and then someone gives you a book this thick, right? Book A is this thick, book B is this thick. And I were to say, if you had to guess, which book do you think has more instructions in it? You probably guess book A, right, as opposed to book B. You might be wrong. There might just be bigger font or, you know, extra pictures, blank spaces, whatever the case would be. But your best guess would be the bigger book has more instructions. So anyway, your book gives some examples, including red-green color blindness, hemophilia, and a type of muscular dystrophy. These are all from sex-linked recessive alleles. You can look into it if you want, especially the hemophilia thing. If you're into history, that's a really interesting one. One could argue without hemophilia in the royal blood from inbreeding and all that, that you might not have, we might not be where we are in world history without that. And I would go into detail, but we're out of time, or we're short on time, and you can look it up if you want. Anyway, your book gives the example of red-green color blindness. That is a common sex-linked disorder caused by malfunction of light-sensitive cells in the eyes, meaning, hey, you've got to be, something's wrong there, right? Normally, things work. In this case, they don't. It's mostly the males that are affected, and you'll see why here in a second. Heterozygous females have some defects, but for the most part, they don't. The next slide is going to illustrate what I'm talking about. Can anybody not see the number on that screen? Everybody sees what number that is? Or if you're on your phone, maybe I'll text it to you. What? Yeah, it's a 7. Anybody not see it? If you can't see it, you might be colorblind. But yeah, there's a number 7 right there. Anyway, this is how a colorblind test goes. Because these genes that are responsible for this are located on sex chromosomes, sex-linked genes exhibit unusual inheritance patterns. So remember, we talked about a pedigree before. Now we're kind of going back to it, even though I'm not going to show you the pedigree. It's still, you know, we don't even have time to talk about it. It's not that important. But here, let's look at this. Here we have, wait, what's this up here at the top left? Is this male or female at the top left? Female, good. It's an X and an X. And then to the right, we have a male, an X and a Y. Now this female has 2 normal alleles, so she's good to go. She has normal vision. She doesn't even have, she's not even a carrier. This male has only one of the abnormal alleles, right? Because it's only on the X chromosome. So because of that, even though he only has one copy, this is what makes this part interesting and what you need to know. Even though we're talking about something recessive, prior to this, I've always said for recessive, you had to have both alleles, right? In this case, we're adjusting it. Rather than saying you have to have both alleles for you to have the recessive phenotype, let's adjust that. And we are now say, if 100% of your alleles have the recessive phenotype or genotype, then you will have the recessive phenotype. Because yes, in this case, the male only has the one allele, not 2, but there's only one anyway. So 100% of this person's alleles are the defective one. So anyway, this person produces eggs. You know, no matter what, 100% of them are going to be X with a big N. This person produces sperm. 50% of them are going to be X with a little n. 50% of them are going to be a Y. And basically what we can see here through this Punnett square, which you might have to do for the exam, I might give you all this information, and you do the Punnett square. And I would say, what are the chances of their kids having color blindness? The answer is 0. And that shows you why. It's a different scenario if the female is a carrier. Whoops. Yeah, you can't see that, but the female is a carrier. She is no longer homozygous dominant. She now has that one recessive allele. She's okay, though, because she has one good allele, but she passes along that recessive allele. So because of that, she now has a possibility of her kids having color blindness, even with a completely normal male. And then, of course, if you have a carrier female and an affected male, you know, you've got a 50% chance of your kids having it. You do not need to memorize any of this. If anything, just know that I might give you this information and ask you to do a Punnett square. Again, hemophilia is a great example. You can read about that if you want, but it won't be on the exam, so we will end it there. Woo. And then the last word for those of you online is going to be lawnmower. So those are 3 words. Make sure you email me to let me know. I'm going to do what I've used, what I always intend to do, which I'm going to post this video as soon as it's available. And then if I get a chance, I will make questions. So as long as you send me the email, And as long as you are Ray, Lloyd, Johnson, Davis, Baumgartner, Fitzpatrick, or Hamlet, then you can send me an email saying, hey, I was here. Here's the keywords. If you're not on that list of people, then sending me the keywords won't matter because you weren't here watching it. And that means someone just gave you the keywords. So anyway, you guys have a great day. I'll be at office hours if anybody needs me.